All right, today we are diving into the world of Earth's ultimate survivors, the Archaea. We're going to crack open the cellular toolkit that lets them live in places so extreme they might as well be on another planet. So, what's their secret? How do they pull it off? Let's get into it.
Okay, so picture this, a boiling, acidic hot spring. You'd think, no way anything can live in there, right? Well, you'd be wrong. This is the world of the Archaea, the absolute masters of extreme survival. So the big question is, what makes them so incredibly tough? What kind of molecular hardware are they packing that lets them thrive where pretty much nothing else can? To figure that out, we've got to start peeling back the layers.
So, our investigation begins right at the cell's front line, its plasma membrane. And look, this isn't just some passive container. It's the critical barrier standing between life and a seriously hostile universe. Now, on the surface, it might look like any other cell membrane, but underneath, its chemistry is totally, and I mean brilliantly, different. So check this out. On the right, you've got your standard bacterial membrane, and on the left, the Archaeal version. The real secret sauce is in the chemical bonds, the glue that holds them together. See, bacteria, and actually all other life, including you and me, use something called ester linkages. But Archaea, they use something way tougher, ether linkages. Now that might sound like a super nerdy detail, but trust me, it is an absolute game changer when it comes to durability. This table really lays it all out, the three key differences that basically rewrite the rules. First up, that ether linkage we just talked about, it's just chemically stronger than the ester one found in bacteria and our cells. It can handle heat and chemical attacks that would just shred a normal membrane. Second, instead of using straight, simple fatty acids, Archaea build with these branched, more complex units called isoprenes. And third, get this, even their glycerol backbone is a mirror image of what everyone else uses. It's kind of like the difference between a left-handed and a right-handed glove. They are working from a completely different set of blueprints. And here's the payoff for all that unique chemistry. Those branched chains can actually fuse together, forming these super long molecules that stretch across the entire membrane. What does that do? It creates a single, rigid monolayer, not the two flimsy layers you see everywhere else. Think of it less like a soap bubble and more like a tough, heat-resistant wetsuit. It gives the cell incredible stability, especially in crazy high temperatures. Okay, so we've got the membrane covered. Let's move one layer out to the next line of defense, the cell wall. And hey, if you thought the membrane was unique, you haven't seen anything yet. The Archaeal cell wall is just a masterclass in adaptation. There is no one-size-fits-all approach here. It is all about building custom armor for whatever job needs doing. So the most common design you'll see is this really beautiful, almost crystalline structure called an S-layer. It's basically a sheet of interlocking proteins that creates the cell's outer armor, kind of like microscopic chainmail. It's simple, elegant, super efficient, and for a lot of Archaea, like this salt lover halobacterium, it's all the protection they need. But of course, some environments are even tougher, and they demand more reinforcement. So what do? Well, organisms like Methanospirillum here just take that S layer and they add another layer right on top, a tough protein sheath for extra support. It's a totally modular design, just adding reinforcement exactly where it's needed. And then you've got this fascinating twist. Some Archaea build with a polysaccharide layer called methanochondroitin. Now, what's just mind-blowing about this is that its chemical structure is incredibly similar to a key part of animal cartilage. I mean, think about that. It's a perfect example of convergent evolution. Two completely different branches of life independently coming up with a very similar solution to the same structural problem. And finally, we have something called pseudomurein. Now, at first glance, it looks almost exactly like the peptidoglycan that makes up bacterial cell walls, but it's a clever fake. It's built with different sugars and different chemical bonds, and that little switch-up makes it completely immune to things like lysozyme. That's an enzyme our own bodies use to blow up bacteria. It's just another brilliant piece of molecular defense. Okay, so we've looked at all this amazing armor, but how does a cell actually build it? And even more importantly, how does it replicate itself? How does it flawlessly construct all these complex layers from scratch while splitting into two? Well, let's dive into the dynamic process of how you build a brand new cell. This is really the fundamental challenge for pretty much all cellular life. If you're going to divide, you have to build a brand new wall right down the middle. If you're off by even a few nanometers, it could be a complete disaster. So how in the world does it pull off this incredible feat of molecular engineering again and again without messing up? The answer is this amazing piece of molecular machinery called the Divisome. The best way to think about it is like a construction crew made up of more than a dozen different specialized proteins. And they don't all just show up at once. It's a very precise operation. First, the early components arrive to mark the construction site, kind of like a foreman. Then the late components come in to do all the heavy lifting of actually building the new wall. Okay, now let's not get bogged down memorizing a bunch of protein names. Instead, let's just focus on what they do. The whole process kicks off with one key protein that forms a ring right in the middle of the cell. It's like a measuring tape, finding the perfect spot to divide. Then, other proteins come in and bolt that ring to the membrane. Meanwhile, another one is making sure the duplicated DNA gets pulled apart safely to each side. Finally, a scaffold goes up for the wall-building crew to work on, and a trigger protein gives the go signal for the final pinch that splits the cell in two. It's just a perfectly coordinated team effort.
All right, let's zoom in on the main event here, the actual construction of that new wall. Now, to get a handle on the basic principles, we're going to use the bacterial process of building peptidoglycan as our blueprint. And it's really important to remember that Archaea use different materials, like that Pseudomurein we talked about. But the bacterial process gives us a fantastic look at the fundamental logistics. How does a cell build something so complex on the outside of itself?
The whole operation is a huge logistical puzzle. I mean, think about it. All the building blocks for the new wall, they're made inside the cell, in the cytoplasm. But the wall itself is on the outside of the membrane. So the cell has to figure out an efficient, totally reliable way to get those building blocks across the barrier and assemble them in exactly the right spot.
And here's how they do it. It works just like a molecular assembly line. Step 1, the basic parts, the precursors, are made inside the cell. Step 2, a key piece gets loaded onto a special carrier molecule. Now this is the cool part. That carrier physically flips the building block right across the membrane to the outside. Once it's out there, it gets attached to the growing wall. And finally, the whole thing gets reinforced with crosslinks to make it super strong. It is pure precision engineering, but on a molecular scale.
So when you put it all together, from their uniquely bonded membranes to their diverse custom-built walls and the incredible machinery that assembles it all, Archaea have this truly remarkable toolkit for survival. And this precise molecular construction, it's the very foundation of how they can replicate and thrive in the most brutal environments on Earth.
And all of this leaves us with a much bigger and honestly more fascinating question. What the Archaea show us is that the blueprints for life are way more flexible than we ever thought. So if their unique toolkit allows life to exist in Earth's most hostile spots, what does that tell us about the absolute limits of life itself? You have to wonder, could similar or even totally different alternative chemistries be supporting life on other worlds right now? It really pushes the boundaries of what we even think is possible.